Good afternoon to everyone. I apologize for the delay. We had some technical problems, so sorry. And so I'm Claudia Paniga, and I have been working for GFB for 4 years. Now I would like to introduce you Dr. Carla Sanchez, who has been working for GFB for a long time, and he will speak about himself and about our host today, Volker Straub. Thank you so much for joining us. Thank you so much for the mission. Thank you.
Okay, welcome. And first of all, I want to say if you can, the organization can put the video on for the speakers, it should be very nice. If not, we will go further like that. It is possible to have a video. No, not now. Okay, so just I continue. So welcome everybody in the name of GFB Onlus to our second webinar. This webinar is called Overview of Patient Registries for Limb-Girdle Muscular Dystrophies and Updates on Ongoing Gene Therapy Projects. We are very happy to have you here today, just in this particular day. That today, if you don't know, it's the International Day of Neuromuscular Disease. And so we are very happy to have you all here. And first of all, I want to excuse also our president, Beatrice Bola, that today cannot be here. And then I present myself. So for those that didn't know me, my name is Carlos, and I am a member of GFB. I am the responsible of scientific affairs of GFB. Our organization is a patient organization that was born in 2013 with the aim to give support to the research. Since then, research has developed at the giant steps. And the more the research develops, the more accurately disease can be diagnosticated. And then the more complicated is to know each one of them. This knowledge that cannot be learned in the books is living in each one of us. And this is important to share this knowledge with doctors and registries to build together the road of our treatments. To treat this topic, we have invited Dr. Volker Straub. He has trained as pediatric neurologist at the University of Düsseldorf and the University of Essen in Germany. He wrote his PhD thesis on Duchenne muscular dystrophy, and he worked as postdoctoral research fellow in Dr. Campbell's laboratory, focused on limb-girdle muscular dystrophy. Volker has long-standing interest in pathogenesis of genetic muscle disease, and his current research is also involved in the application of magnetic resonance imaging, next-generation sequencing, and other omics technologies that for the characterization of primary neuromuscular disorders. Currently, Professor Straub is the Deputy Dean of Harold Macmillan Professor of Medicine and Professor of Neuromuscular Genetics at the Institute of Translational and Clinical Research at Newcastle University in the UK. He's the Director of University's John Walton Muscular Dystrophy Research Center and holds honorary clinical appointments. Welcome, Volker, and thank you very much to be with us today. And the floor is yours.
So thank you very much. I'm not entirely sure if you can hear me.
Yes, yes.
Okay.
We can hear you. I don't know if the video is currently working or not, but yeah, we can just hear you.
Okay. And so it doesn't seem that you can see me, so I can't activate my video. Sorry for this. I can also currently not share any screen, any presentation, because it said who then changed the nomenclature and defined limb-girdle muscular dystrophy new. And what we decided is that they should be classified as D for dominant and then R for recessive, and then we add just a number instead of a letter in terms of their historic description. So R1, alpine related, is the most frequent form and was the first one described. R2 is actually the second most frequent one and was the second one described. And so depending on when they were described, they get this number. And we now have actually already more than 24 of the recessive one, and there are 5 from the dominant ones. I don't want to go into the genetics. There are new diseases that we have now described. This is why the list is longer, and I'm fairly certain that there will be even more forms of limb-girdle muscular dystrophy in the next year. So I'll just give you one example of the work that we do. So we work in Newcastle. I live in Hexham, which is a small town outside Newcastle. And in our cathedral that we are very proud of, this is Carsten Bönnemann who is there, you can read that the north of England was actually conquered by the Vikings, and some of you may have also seen the Netflix series, The Last Kingdom, or the Vikings. So the Vikings came to Northumberland and to England. Interestingly, the Vikings also went to Italy and to the Mediterranean Sea. And so this was something that you could see in a number of movies. And so the Vikings also left their genes there, and there are a number of genes with limb-girdle muscular dystrophy that actually are coming from Scandinavia. And in our that is the most frequent forms are actually those forms of limb-girdle muscular dystrophy. For us, the most frequent form is LGMD2I or R9. In Italy, this is less common. And in Italy, the calpain 3 is the most common one, and then dysferlin. The sarcoglycans, of course, are very well characterized, and there are also many sarcoglycan families in the Mediterranean countries, more than for FKP or anoctamin 5. What's very important for us is to understand where patients come from, because different countries have different forms of limb-girdle muscular dystrophy when it comes to frequency. We studied these in a big project where we did genetic testing across 50 centers in Europe. It's a project that is called MyoSeq. And so we collected DNA in the centers, and we then did the genetic analysis and provided the centers with a diagnostic report. We diagnosed the patients also including, clinical information, also sometimes MRI and muscle biopsy. And I don't want to go into the project in too much detail, but we have provided more than 1,000 patients now with a diagnosis with limb-girdle muscular dystrophy. And in Europe, the most common form is LGMD1 or calpain 3. The second most common form is dysferlin or LGMD2, and then anoctamin 5, and then the sarcoglycans. So we still have a lot of patients that don't yet have a diagnosis and are unsolved. But this again shows you that calpain 3 or LGMD1, formerly called LGMD2A, is the most common form in Europe. So this gives you an idea about the patient numbers. And again, it's just summarizing again, the most common forms are those 2, R1 and R2. But it's also quite common more in the north of Europe that we find LGMD2L or now R12 caused by Noctamin 5. So I, sorry, just want to summarize this again in this slide, what the most common forms are. Now, when we compare this with other studies, we only looked in Europe. This is a large study in the United States. There also calpain was more common, or a little bit more common than dysferlin, but and then FKRP was very common as well, like in the north of England. So a lot of this genetic testing is now automated by computers, and it's very important that every patient with limb-girdle muscular dystrophy does actually have a specific diagnosis, because that is important for care, also for registering in patient registries. And so these are registries, and then there are several more now for limb-girdle muscular dystrophy patients. So the Coalition to Cure Calpain 3 has a registry for LGMD-R1 or for calpainopathy patients. Then the Jane Foundation has a registry on their website for dysferlin or LGMD2B, now called R2. And then there are, of course, patient registries for the sarcoglycans, also the Intelligent one, of course, for beta-sarcoglycan. There's an FKRP registry that we host here. So there are quite a number of patient registries. I haven't listed them all here, but I think it's a really helpful tool. We actually used one of the registries, our global FKRP registry, to also provide patients with newsletters about care and about research. We are now collecting more information about limb-girdle muscular dystrophy in projects across Europe and the United States. And so there's a big consortium called the GRASP Consortium that is looking at a number of limb-girdle muscular dystrophies and to prepare them for trials. So in Newcastle, we have done this particularly by assessing patients by physiotherapists and also by magnetic resonance imaging. We collected a lot of data for patients with LGMD-R9 or 2I. And what was interesting for us is that the biggest change was in terms of breathing rather than in terms of weakness. So the respiratory change was really the only thing that we found that changed over a 12-month period. Even after 6 years, some of the patients were still improving when they were walking. And so, it was a lot that we still need to learn. I think for many of the limb-girdle muscular dystrophies, we still need to have more data. Now, just to say that we are now starting to also have new treatments for limb-girdle muscular dystrophy. So for limb-girdle muscular dystrophy R9 or 2I, there's now a gene therapy study that is sponsored by Genethon and a small company from Genethon called Atamayo. So the first patient has now been dosed with gene therapy for this specific form of limb-girdle muscular dystrophy. The other group that I want to briefly introduce is the dysferlin patients, or LGMD-R2. So we have collected data from more than 200 patients across from 15 sites in the world. And this is the so-called clinical outcome study for dysferlinopathy funded by the Jane Foundation. And these are all the assessments that we do for motor performance, and we test how well patients can rise from the floor or walk. how strong they are, how good their respiratory function is. We'll also look at quality of life. And so this study is still ongoing. We have now extended this also to South America and to South Korea. And we know that these studies where we just collect data to understand the disease will help to also then coordinate clinical trials. So we expect that there will also be clinical trials probably next year for dysferlin or LGMD2. And then for the sarcoglycanopathies in particular, Sarepta Therapeutics, a US-based company, has now started to look into gene therapy. There's a first clinical trial for LGMD2E or beta-sarcoglycan deficiency from Sarepta, and they are planning to do more gene therapy studies for alpha-sarcoglycan and gamma-sarcoglycan, but in the future also for dysferlin, anoctamin 5, and calpain 3. So there is a lot happening in the field. It's a very exciting time. It's important that every patient is having a diagnosis, and the diagnosis is a prerequisite often to actually be included into clinical trials, but also into patient registries. I just want to acknowledge some of the funders that have funded and supported our work, and colleagues across Europe that have worked with us on the diagnosis of limb-girdle muscular dystrophies. These are some of the colleagues in my own lab. So there's a lot of work to do, but it's a very exciting time for patients. I also want to thank actually patients and the Jane Consortium. And I want to thank you for your attention, and I apologize that I didn't specifically provide more information about the registries, but I'm very happy to, of course, have questions and answers now. So thank you very much.
Okay, thank you, Volker. And now I saw that I still didn't activate my video, but during the registration, we have had some questions that we will give to you. And now, if some people of the audience have some question, you can write it in the chat, and we will step by step do these questions to Volker.
Thank you.
For the moment, I just start with one question that is a person that say that she have a son of 15 years old with a limb-girdle muscular dystrophy 2E, so beta-sarcoglycanopathy, that progress rapidly. And they are looking forward for gene therapy. And the question is, if gene therapy will is hope to stop the disease, it can stop the disease. She say even if it will be possible to recover after the therapy. And the question is as well what to expect of these gene therapies. So and how we can maintain now our bodies to be as strong as possible until we wait for these therapies.
Yeah, so these are all very important questions. Maybe starting with the last one. How can you try to maintain your current motor function and mobility as good as possible? Well, unfortunately, there is nothing that one can really do to stop the progression of the disease, but in terms of mobility, particularly when it comes to contractures, it's important to see a physiotherapist to make sure that the mobility of your joints is as mobile as possible. But But in terms of weakness of the muscles, there isn't really anything that you can do to stop the progression. I think it's also important for the sarcoglycanopathy patients in 2E to make sure that you have had cardiac assessment and make sure that your cardiac function is good. For some of the gene therapy trials, it is requested that you have a cardiac function in a certain range. So therefore really important to look at the heart function and get medication if possible. And that is relevant for clinical trials. Now, what to expect from the clinical trials? We do clinical trials because we can't predict what to expect. The question also was, will the gene therapy be able to fully recover your strength? Most likely not. Some of your muscles in the sarcoglycanopathies and other limb-girdle muscular dystrophies are completely replaced by fat over time. So if the muscle tissue is replaced by fat, then the gene therapy can't do anything. So I think the best that we can probably expect is that the gene therapy will stop the progression. Where the muscle is still very good, for example, in the arms or hands, it might be possible to improve function. But where the muscle is gone, I think you can't really improve anything anymore. I hope that this answered it, but yes, it's a very complicated question. There's a lot to discuss.
And this I add by my own, it should be possible then to combine some other treatments with gene therapy. So I mean, maybe in gene therapy, you can stop the disease. Then maybe we can expect or we can hope in another treatment to just, for example, as you mentioned, the fat, some drug or something that can convert the drug again to the muscle, or this is science fiction?
No, I do think that it might be possible to also combine gene therapy with other treatments, absolutely. So there's some companies now working to also induce muscle growth with myostatin inhibition. Then there are other companies that try to reduce the amount of connective tissue. Then, of course, there are many patients with sarcoglycanopathies that have used steroids particularly when they were younger. So I think it will be a combination of treatments in the future. The big question for patients is always, well, when does this future start? And that is currently difficult because it takes a long time to get a drug to the patient, particularly if patients don't participate in clinical trial. So I think it will happen that there will be many drugs, but right now it's still very challenging.
Mm-hmm. Okay. So then another question that it always comes to our minds is that there are a lot that have been developing in Duchenne muscular dystrophy, and someone asked if these progresses in medicine for Duchenne will also help our diseases, other dystrophies.
Yeah, that's a very good question. So Duchenne is the most common form of muscular dystrophy, and it's, of course, a very progressive and very severe disease. This is why there are more clinical trials and more companies. But so far in Duchenne muscular dystrophy, there aren't any treatments that have been approved around the world. So there are now 2 new trials that have shown good results. One of them is for a drug that is called vamorolone, and another one is for an Italian company that is called Italfarmaco, and the drug is Givinostat. They have both shown to be helpful in Duchenne muscular dystrophy, and I don't see any reason why they shouldn't be helpful for limb-girdle muscular dystrophy. And the companies are now thinking about also doing a clinical trial for limb-girdle muscular dystrophy. It's important to mention that if a drug works in Duchenne muscular dystrophy, the pharmaceutical companies often will only get a license for Duchenne muscular dystrophy. To get a license then for limb-girdle muscular dystrophy, they may still need to do a clinical trial to show that it's also working in limb-girdle muscular dystrophy. But generally, I think any research in Duchenne is also good for the limb-girdle muscular dystrophies.
Good. Okay, so another question may be related to the thing that was we spoke before is if supplements. So we have heard about supplements and integrators, and this is the supplements help and how to choose them.
Yeah, so for many supplements, often these are vitamins, for example, there aren't any clinical trials. When you live in the north of England, then, for example, vitamin D is very important because we don't have enough sun. Whether if you live in Italy, you probably don't have any vitamin D deficiency. So there is no supplement that we can say definitely helps. People might have heard about creatine, which is a supplement that bodybuilders also use. And creatine has been tested for patients with muscle diseases, and it might show some benefit. So it's something that one can try. Often the health service doesn't pay for it, so you would need to pay for it yourself. I also saw that there was one question in the chat about growth hormone for calpain 3 deficiency. I would advise against growth hormone because growth hormone has quite a lot of side effects. It's very expensive. The concept that it is a so-called anabolic drug is correct, but I think it's something where there is no evidence that it would work in a patient with calpain 3 deficiency.
Okay, so another question. There is one woman that say that he have read about the SMA, that not all children with a diagnosis are suitable for gene therapy treatment, even if they have the diagnosis. And his question is, for whom could be also suitable a gene therapy, for example, for beta-sarcoglycanopathy or other sarcoglycanopathies, even if they have the diagnosis. So
yeah, that's a very important question. So the gene therapy is currently delivered by so-called viral vectors. So to get the gene into the muscle cell, researchers and pharma companies use viruses that transport the gene to the muscle. Now, these viruses aren't dangerous, but many people have so-called antibodies against these viruses. So this is very similar to vaccination, or if you have had an infection. Everyone now knows COVID. So if you get a COVID vaccination, you then produce antibodies that will hopefully defend the virus if you get it. This is the reason why gene therapy in spinal muscular atrophy, for example, cannot be given to everyone, even though it is approved. But if you have antibodies against the virus, then the treatment will not work, and you may actually have an immune response, a very strong one. So therefore, every patient will be tested for antibodies against the viral vectors before gene therapy. And in limb-girdle muscular dystrophy, there might be, I don't know, in some regions, 50% of the patients that have antibodies. So even if gene therapy for limb-girdle muscular dystrophy would work, it doesn't mean that everyone would be eligible.
Okay. Other questions are related to the treatments against anactomine 5 and calpain you say again, and gamma sarcoglycanopathy. But they are just asking what is new in this disease.
So yeah, so for anactomine 5, I think what is new is that many centers are now working together to collect more information about the disease symptoms and the disease cause, which is very helpful because right now there is no interventional or drug trial, but it's good to do natural history studies. For gamma-sarcoglycan, Sarepta Therapeutics, the company in the States, is developing gene therapy. This needs to be tested in a clinical trial. In the future, also for anactomine 5. Now, for calpain 3, there are actually several initiatives where also gene therapy will be tested. One is in France by Genethon, but there are also researchers in the United States who are looking into gene therapy treatment for calpain. I need to say that none of this is yet available.
Okay. And another question, it is how can it be possible that two brothers with the same genetic mutation, the same parents, have different outcomes of the disease? No, maybe that one being more severe than another one, or with different muscles affected.
Yeah, this is an excellent question and really interesting. So we see this in many diseases, that there are differences between siblings, between sisters and brothers, or brothers and brothers, or between different generations. The reason for this is that we are all different, and we do not know right now why or what the responsible genes are that can modify the symptoms, so make them better or more or worse in one brother compared to the other. So we don't have a good explanation. We just know that, yes, this is the case, and it's probably because we are all genetically different. By learning more about this, we may be able to also think about treatments. So we know that even in identical twins, you can show differences in the severity of symptoms. And it just shows that it is more complex than just a single gene. but a very interesting question, and there's no good answer why this is the case.
And perhaps the next part of this question is not just between siblings or brothers or that, but inside the same person. No, all limb-girdle muscular dystrophies show that some muscles degenerate very fast and some muscles are resisting. There is an explanation for that, or
there is no good explanation for this. And it's a very good question. We don't know why some muscles are more severely affected than others. What we do know is that this is not just because of the mechanical use of the muscle, but there's probably also genetic differences between different muscles. There are differences in the metabolism of different muscles, without going into too much detail, but we also have muscle fibers that do different types of work. Some of them are more for sprinting and others more for endurance running. And all of this might play a role and explain why different muscles are showing different degrees of involvement.
Okay. Now we are arriving to the end of our questions. Maybe I let some question here in the chat because I could not read it all. So if there is some question that...
Yes, Carlos.
Okay, say it, Claudia.
If you want, I can read. Okay, can you hear me?
Yeah.
Yeah, okay. So actually, I put together two questions because they are about clinical trials, one for dysferlinopathy and the other one for 2G. So they want to know how is the situation at the moment and what the situation will be in the future, mainly. So these two subtypes.
Volker, your microphone.
Yes, thank you. So yes, for dysferlin, I'm very confident that we will see first clinical trials probably next year, more towards the end of the year. So Sarepta is interested in a gene therapy study in dysferlin, but we have to wait also to see how safe some of these gene therapy studies are. And then for 2G, so this is telethonin. Now, the problem with telethonin is that so 2G is an extremely rare form of limb-girdle muscular dystrophy. I'm currently not aware of any planned studies, but what I do know is that group in the United States, in Columbus, in Ohio, are looking also into gene therapy for 2G. But this could still take a while, but at least there's an interest. I know that they're doing research on that.
Thank you. I've got other questions. Sorry. How to apply for testing in the future?
So when it comes to genetic testing, I think the most important thing is to talk to your neuromuscular doctor or make sure that you are seen in a center that has access to genetic sequencing. I would say that almost all patients with limb-girdle muscular dystrophy should have a genetic diagnosis, and it should be possible to just have a blood test, and that you don't need necessarily a muscle biopsy anymore. So the advances are very striking, that we are very good now in making a genetic diagnosis. So it's just a blood test, really.
Okay, so another question. Did you hear about the HMB supplement? Have you ever heard about it?
So I saw this in the chat, and I'm not entirely sure what HMB stands for. So sorry, I can't answer that question.
Okay, and the last question I have is, what about the influence of corticotherapy in LGMD2C?
Oh, just a sec. Yeah, I just checked what the HMB stands for. So I can briefly come back to this. So
Okay.
because this is hydroxy methylbutyric acid. So this is a supplement as well that has been used actually in patients with muscle disease. It hasn't been tested systematically in limb-girdle muscular dystrophy. It has been tested in spinal muscular atrophy and also in Duchenne muscular dystrophy. There's an idea that this could work, but right now I couldn't advise anyone on taking it because there's no evidence that it works in limb-girdle muscular dystrophy. And then I think your next question was about steroids in LGMD2C. So this is for gamma sarcoglycanopathy. I think if you are young, if this is in a child with a severe progressive disease, I would recommend steroids. It's also important, I think, that most of our experience is from Duchenne muscular dystrophy, so from boys. With females, particularly when it comes to puberty and having children, steroids might not be the best. But generally, there is some evidence that steroids can improve muscle function in sarcoglycanopathies, particularly in the more severe forms.
Okay, thank you very much, Dr. Straub. I don't have any other questions. I can't see any other questions. So I thank you so much for being with us. And I hope that this, I'm sure it was helpful for all our patients. And thank you very much for being with us. And thank you to Dr. Carlos Sanchez. And thank you to everyone who attended this webinar. Thank you. Bye-bye.
Thank you very much. So much. And apologies for being late.
Don't worry, it's okay.
Bye.
Bye-bye. Bye. Bye. Bye, everyone. Bye-bye.
Bye.
Bye.